Welcome to the fifth talk in the webinar series on genetics and evolution, organized by the Journal of Genetics. I'm very happy and honored to introduce Professor Laurence Mueller. He works at the Department of Ecology and Evolutionary Biology in the School of Biological Sciences at University of California, Irvine. Professor Mueller got his PhD working with Professor Francisco Ayala at the University of California, Davis, working on density-dependent selection. For his postdoctoral work, he worked with Professor Mark Feldman in Stanford University, where he addressed a very interesting problem working on the population genetic theory of kin selection, extending the idea to multiple alleles and true loci. Since 1988, he has been working at University of California, Irvine. Professor Mueller is one of the pioneers of the modern experimental evolution approach. He established many of the tenets of this approach during his PhD research. He is especially known for his theoretical and experimental work on density-dependent selection and adaptation to larval crowding, especially using the fruit fly system. Along with his colleague, Professor Michael Rose, he has greatly improved our understanding of how the shapes of age-specific mortality and fecundity curves evolve and why aging stops at advanced ages. In addition to all this, he has also had a great influence on the sciences of ecology and evolution through the people he has trained. A large number of evolutionary and ecology labs, especially in the Indian context, trace their ancestry back to Professor Mueller's lab at UC Irvine. Today, he will be speaking to us about his most recent research passion, mapping genome transcriptomes to phenotypes. Professor Mueller.
Thank you, Prasad, for that nice introduction. I'd like to start because sometimes we save these to the end and we have to rush it by acknowledging my collaborators. Of course, probably the most important is Professor Michael Rose, who's seen here in this photograph, who I've known for almost 40 years and have actively collaborated with for probably 30 of those years. Many of the populations you'll be seeing that with experimental results were derived in his lab. The next two people, Dr. Barter and Kasia Bittner, are recently graduated students from my lab. Dr. Barter helped with the transcriptomic work and some of the phenotypic work that I'll show. Kasia Bittner did a lot of the work that I'll show on urea-resistant populations. Dr. Mark Phillips was a former student of Michael Rose's. He helped me with some of the statistical work on this method called FLAM that I'll introduce in a second. And finally, Mr. Zach Greenspan is my current graduate student who also helped out on the statistical work that I'll talk about. I do work in aging, and one of the things I'll tell the younger people watching this talk is that there are many features of aging, like getting gray hair, wrinkled skin, telling the same story over and over again. But another feature of aging is you start to use photos in public of yourself that are at least 20 years old, like the one that I've shown here, where Dr. Rose is in the foreground and I'm in the background. In any case, what I'd like to do first is start with a summary of the kinds of populations that we do experimental evolution with. This is an outline that highlights some of the selection regimes used by Michael Rose over the last 30 plus years. And we'll see some of these populations as we go through this talk. So one set of populations that I'm showing here are called the A types. They're selected for accelerated development, and they're on a 10-day life cycle from egg to adult. We then have another set of populations called Bs, which are allowed to develop a little longer, 14 days, when they are then allowed to lay eggs and start the next generation. There's another group of populations called the C type, which spend a more extended period as adults, up to 28 days from egg before they reproduce. And then one of the most, the longest lived populations are called O's. They eventually had their life cycle stretched out to 70 days from eggs. And some of the populations you'll see that we use are derived from the O's, although we don't do work with them anymore. I also have a series of populations that I've listed here called the UX, UTB, and RUX. These populations have been kept in larval food that has urea added to it, which is typically a toxic nitrogen product, but over time they've evolved resistance to urea. And the UX, UTB, and RUX are on a 21-day life cycle. All of these selection regimes are at least fivefold replicated, and I'll show a little more of the phylogeny of these in the next slide. This is just actually a partial phylogeny of the lines that we'll be using. Michael Rose started these from a population called IV, which he collected in the late 1970s. His initial two selection regimes were called O's and B's. You can see next to each name for a selection regime is the number of generations that these populations have been under the particular selection as of November 2018. And of course, since the B's are on a 2-week selection regime, they've undergone many more generations in that environment than have the O's, which are now on a 70-day life cycle. From the O population, for instance, the COs were derived, and they've now been in their environment for 343 generations. But as you can see, there's a new version of the COs called the NCOs, which were derived more recently. They're also fivefold replicated. They've been under selection for 89 generations. And we have a number of populations like that. For instance, there are also, there's a population called ACO, the Accelerated Development Line. That's now been under selection for 884 generations, but a newer accelerated line called AOs, derived from the O's, has been under selection for 293. Down towards the bottom of this figure are these urea-selected lines, UX, RUX, and UTB, along with their control, AUC. The RUX are different from the other two in that for now about 100 generations, they were moved back to standard food. Therefore, they've been under relaxed selection for 100 generations. Now, one of the things that happens when you put populations in very different environments like this is that they become differentiated for a number of phenotypes. I want to illustrate this with just a few of the phenotypes we've looked at. On the left-hand figure here, we have development time from hatching until the fly closed as an adult. And if you look down to these three figures, you can see that the development times are are very early for the A lines, right, because they're selected for rapid development time. The other thing to notice is that even though the ACO and AOs have been under selection for a very different period of time, they seem to converge, which probably isn't surprising given that even the AOs have been under selection for over 200 generations. The B and BO, which are given more time to develop, have a slightly longer development time that you can see because these three graphs are on the same X coordinates. And then finally, the Cs versus the COs are the most slowly developing set of populations at the bottom. And again, all the different versions of Bs and Cs have converged on the same development time profile. So that's one phenotype that differs among these populations. We can look at early fecundity, which is shown on the right figure here. The green points are for the AO and ACO populations. These are the rapid developing lines, and you see that they start reproducing or being capable of reproducing very early, and they reach a peak very early, and that starts declining. The blue dots are the NCO and CO populations, and they actually take much longer, 40 to 60 hours longer to start reproducing, at least the females, and laying eggs, although they eventually are a little more capable than the accelerated lines of producing eggs. So that's one set of phenotypes. Here's another phenotype for that same set of populations, the A's and the C's. This is adult mortality, so the y-axis is the log of mortality. This is for adults as a function of their age. Males on the left, females on the right. What we can see is that the accelerated lines start aging very quickly, and a straight line on this graph represents a Gompertz type of mortality trajectory. So the accelerated lines are aging quickly. The C lines, which are the blue dots, which reproduce later in life, in fact, they reproduce again at 28 days. You can see that the mortality rates in males and females are almost constant until day 28, and that's about when the lines start, that's when the lines start to increase in a linear fashion, actually exponential. All of these populations also tend to show a leveling off late in life. Those are mortality plateaus, which I'm not going to talk about today. Nevertheless, there are very different phenotypes between the A's and the C's. And in fact, what is we'll look at later is if you look at mortality at day 21, you can see there's big differences between the A's and the C's. And one of the genetic studies of RNA levels was done at day 21 in these populations. So the point here is that the selection that we apply to these populations doesn't change just one phenotype, it changes a multitude of phenotypes. And one of the things that we're ultimately interested in is discerning what genetic changes are responsible for changing these phenotypes. I'm moving on now to another set of populations just to show you what adaptation to urea looks like. What we did in this experiment is 50 larvae were put in either standard food without urea or food with about 21 molar urea. And we subtracted the number of survivors in the control vial. We subtracted the number of survivors in the urea vial from that. And so a large positive number here means there was a very big difference between survival in the control environment and the urea environment. And these first four populations, TSO, AUC, CO, NCO, are naive populations that are not adapted to urea, and they show a big difference in their survival in the control and urea environment. The RUX, UX, UTB, the last three selection regimes, are all populations that have adapted to some extent in urea, and you can see that they have, there's a much smaller difference between their survival and control in urea food, as if they're barely affected by the urea at all. So that adaptation is obvious in their ability to withstand high levels of urea. So again, these populations have a number of phenotypes that are different too. One phenotype I'm not going to have time to show that differs in these populations is the rate at which the larvae feed. The larvae that feed in the urea food feed much more slowly than the other larvae, and that's a phenotype we'll be doing genomic tests on towards the end of this talk. So if we want to study these populations genetically, how do we go about doing this? I basically stolen the figure on this slide from Christian Schlotterer and his colleagues that shows a very useful method called pool sequencing, or pool-seq for short. And in this particular little figure, what he's basically showing is on the top graph, we have populations that are through time undergoing some sort of selection. I think in this figure he was talking about ionizing radiation, but it doesn't really matter. So if you move from left to right, you're going from the start of the experiment to many generations in the future. The second line, labeled B, shows what happens in the population to genetic variants. So initially, there's a rare genetic variant that's this dark color, and it has linked genetic variation next to it. It confers some advantage in this new selective environment. So over time, as we move from left to right, that dark variant starts to increase in frequency due to selection. You can also see he's shown that the amount of link variation decreases over time as recombination happens, but that target of selection has increased in frequency. So sometime in the future, we take from these populations a collection of flies, several hundred per population, and we do entire genomic sequencing of that pooled sample. So what we get are shown in this figure, where at each site you have multiple segments of DNA from different individuals that have been sequenced, and this allows you to then compute the frequency of single nucleotide polymorphisms at perhaps several million sites in these populations. Schlotterer is shown on the left, the control population, which has very few of those dark spots. On the right is the selection population. And then we can compare the frequencies of these nucleotides at each site to see which ones are significantly differentiated. And Schlotterer says, at the last step, the causative variants can be identified by contrasting the allele frequencies between base and selected population. Now, that is true, but what we can't tell by just identifying those differentiated loci are which ones affect, for instance, development time versus which ones affect early fecundity versus which ones affect, say, adult mortality. There's no reason to expect that the same set of genetic variants are affecting all of these phenotypes together. So the more difficult part of these techniques is after identifying those sites that have become significantly differentiated, then figuring out which ones are affecting the different phenotypes that show differences among your populations. So I will now switch to the next problem, which is how might we address this fairly difficult problem. So there are, in the experimental data I've shown you, for instance, we have A and C groups, the accelerated development and the more prolonged reproduction lines called Cs. And as I said, there are hundreds, perhaps thousands of significantly differentiated SNPs or single nucleotide polymorphisms that are differentiated, but which ones affect development time versus fecundity versus mortality. So the basic data that we have is a measured phenotype, but it's a measured phenotype per population because the populations are the sampling units, because we are not at the point yet where we can genotype every single fly. So we only have genetic data on a whole population. So we want to take the phenotypes for each of our, say, N populations and see if we can predict those phenotypes from genetic data. And the genetic data might look like this vector, X1, X2, up to XM, where those are the differentiated, those are allele frequencies or nucleotide frequencies at M different sites that we found are differentiated. The problem is that M, that is the number of differentiated nucleotide sites, is much, much greater than the number of populations we have. N might be, 10, 20. In the Rose lab, we can get up to about 60. But M, the number of sites that we're looking at, is in the hundreds. So how do we do that? Well, statistics has a number of different ways of addressing this problem that I've listed out here. Some of them, there's stepwise regression problems where you build more complicated models. There's a set of shrinkage methods which have penalty functions like ridge regression and lasso regression. Or you can try and collapse the variables by using principal components and then using principal components with our regression technique. I mean, these are all things that people try to handle this kind of problem I'm talking about, where you have many more independent variables than you do samples. I'm going to look at a method which is actually a variant of the Lasso, which is called, it has this weird name called FLAM, but FLAM actually stands for Fused Lasso Additive Model. This was first described by Ashley Peterson and some of her colleagues. She's now at the University of Minnesota, and here's the reference for that. So I did not invent this, but found that it was an incredibly useful technique for the problem that I had. Basically, what FLAM's going to do is it's going to create a description of the relationship between SNP frequencies and the phenotype at each locus. So I'm going to call a single nucleotide polymorphism now a locus. So at each locus, it's going to develop a relationship between the allele frequencies and the phenotypes in using a simple step function. And it's going to do that for many different loci, and those effects get added together. So that's why FLAM has the additive part in it. But it turns out, as I said, we have too many loci for the number of populations of independent data we have. So we have to somehow trim the input of independent variables or features, as they're sometimes called. FLAM does this through the use of a group penalty function, where the value of each locus is weighted by its contribution to something like the sum of squares. And if we have loci that aren't terribly informative, they get shrunk down to basically zero, meaning they get thrown out. And only the valuable loci remain from this process. And FLAM, as a result, produces what's called a sparse set of loci. That is a number of loci or SNPs that we'll use, M tilde, and that's much less than our original starting number. And that then gets used to predict phenotypes. Now, so let me go into a little more detail about how the technique works, and sort of zoom through this because, you know, statistics is a useful tool. We're not statisticians, or at least I'm not, but just to give you sort of the flavor for how it works. So, as I said, there's a step function that's generated at each locus. So that step involves minimizing, and I'll point over here, I've written out this minimizing function, something here that's basically a sum of squares where you're contrasting the predictions, that's these thetas, to the actual phenotype. Plus there's this penalty function which adds something to this sum. And this penalty function will be made smaller if everything's zero. Now, the way things become zero is when you create a step function, if you have a flat step function so that adjacent SNP frequencies, and what I've done here is on the x-axis, I've taken all my different populations at this locus and I've plotted them by putting the population with the smallest SNP frequency first, that's X1, the one that has the next largest SNP frequency, that's X2, and so on. And then we have their phenotypic values. And so FLAM is going to create a step function like this to try and follow the actual predicted relationship between the SNP frequencies and phenotypes, but it's subject to this penalty function. And it's only going to make a step if that really helps improve the sum of squares. Now, once we've done this at each locus, we then add it to all the other loci that we're looking at, and the final minimization problem looks like this. So here's the sum of squares term where we're summing over all the loci. Here's the individual fit at each locus. But here's the last term here is the what they call the group penalty function, which is just adding up the value of those parameters. And if a particular locus doesn't make a really important contribution to reducing the sum of squares, then that contribution of that locus basically is shrunk down to zero. Essentially, it's eliminated. Now, this particular minimization problem has two what are called penalty function constants, alpha and lambda. We don't know what those are ahead of time, so the way FLAM works is you basically try a whole bunch of values. Fortunately, alpha and lambda are numbers between 0 and 1, so we can try, alpha is between 0 and 1. Lambda is a positive number, but there's a way to figure out what the upper limit to lambda is. So we have a grid of lambda values and alpha values that we test, and basically the way the program works is it divides up your data into a testing set and to a training set, and then you estimate this minimization function and therefore all these data parameters, and then you predict the phenotype on your test set and calculate the mean squared error, that is the difference between the prediction and the observed value squared, and the values of alpha and lambda that minimize the mean squared errors are the ones that you choose as your as your best estimate. So that's how you get alpha and lambda. Now, it turns out this minimization function doesn't have a unique solution, and we took advantage of that when we implemented FLAM in our in looking at genetic data. So we have a statistical technique. What I described so far is just the sort of outline of the method that Peterson developed. What we have to now do is see if it works with databases that look like our genetic databases. So we did that by creating synthetic databases of genetic data, and the features of those genetic databases that we created are outlined here. So these are like artificial pool seed databases where we're going to apply FLAM and see if we can actually pick out the causative loci for a particular phenotype. So we had a total of 2,000 SNPs in each of these databases. 30 of those were differentiated between the set of populations we looked at. So these would be the kinds of SNPs that would be identified by the techniques that Schlotterer uses. And those 30 SNPs actually contributed to the final value of the phenotype of that population. Now we had another 10 SNPs that also had the same level of differentiation that those first 30 did, but those SNPs made no contribution to the phenotypes. So these are the ones we're really worried about, right? The differentiated SNPs, these are ones that Schlotterer would pick out as, oh, look, they're very different. Selection clearly has worked on them, but that doesn't mean they contribute to the phenotype of interest. So we created 10 SNPs that were differentiated but made no contribution to the phenotype, and then the remaining 1,960 were sampled from a common distribution, and occasionally the random sampling would make them look like they might affect the phenotype, but they didn't also. The 30 causative loci were divided into 3 groups, equally of 10. One set of 10 had a strong effect on the phenotype. Second group of 10 had a moderate effect, and the third group had a weak effect. And this is how the phenotype was computed in these synthetic databases, where these P's are the SNP frequencies, allele frequencies in each of those 3 categories. And then every phenotype has some error term on it due to measurement or environment, which sort of cloud up the signal. So we added different levels of error to our phenotype measurements. Now, the details of this are now published. Here's the reference below for anyone that really wants to stream through all the gory details. But let me just show you a few of the results. The first result, that is this first slide, is looking at the effect of that environmental variation on the phenotype. So let me sort of go through what these figures show, because I'll have a couple of them. On the x-axis are the number of populations that we tested in this artificial database. So we went from a low of 6 populations all the way up to 60. And then we have different lines corresponding to the black figures, symbols, are those causative loci. Those are the loci that actually contribute to the phenotype. The open symbols, the diamond and the triangle, the diamond corresponds to loci that are significantly differentiated but do not contribute to the phenotype. And then the triangles, I'm calling those neutral, but that basically these are the undifferentiated non-causal loci. So let's look at the bottom figure first. you can see a couple of trends. First of all, as we look at more independent populations, our ability to find causative loci increase. Now, the maximum number of causative loci for weak, moderate, or strong is 10. When we have only 6 or 10 populations, we're only catching maybe 2 of those 10 causative loci. But as we increase the sample size, number of independent populations, we get closer to 10, although not at 10. And you can see when this environmental variation is pretty low, that those loci that are differentiated but non-contributing, although they sneak in every now and then, are much lower than our ability, that is, FLAM's ability to find the causative loci. Now, if we increase the noise, which is the top figure, we've increased the standard deviation by a factor of 10, you can see this line with the diamonds increases dramatically. Those are the differentiated non-causal loci, which are now being picked up by FLAM because the environmental sampling noise on our phenotype is starting to obscure the genetically based differences, and that makes it harder for the technique to work. So one issue with the technique is how bad is your measurement error and other sampling variation on your phenotype. Now, as I mentioned before, it turns out that minimization problem that we use for FLAM does not have a unique solution. So Peterson and others that derived this method, and fortunately for me, wrote an R package that will implement it, use one type of numerical method for finding that minimum, which is called coordinate descent. And this particular technique, what it does is it will find one of perhaps several minimum, but it does so by sort of running through the genetic data, which is organized in a matrix where each column of the matrix is a different SNP, and each row of the matrix is a different population. And the coordinate block descent works from the first column of genetic data to the very last column. And it turns out the minima it finds depends on the order of the genetic data. So what we did is we would run FLAM once, we would get one of these sparse sets of loci that were identified as causal, and then we would permute the columns of the genetic data and then run FLAM again. And you would get a slightly different set of sparse loci because, as I said, there are multiple solutions to that problem. And so we used as a criteria for which loci or which SNPs to include as our causative loci several. One was shown here in the top figure, only the loci, and we did that permutation 100 times. So only the loci that showed up in 50% of the most common locus were chosen as causal, or we repeated the simulation choosing only those that showed up 40% or 30% or 20%. And you can see from this first figure that, now this is, again, the number of populations is on the x-axis, the number of causal loci that were identified is on the y-axis. Here we're only looking at the strong causal loci, that as we sort of make our criteria more liberal for what to include, that is, go to 20%, we actually pick up more and more of the causal loci. The problem is that if you look at the second figure, the differentiated non-causal loci also increases as we weaken the selection criteria for what to include. So, so the search for multiple solutions to the FLAM minimization problem can help you identify more of the causal loci, but you have to be careful here because that also increases the chances that you'll get loci that aren't really important contributors. The final thing that we looked at, and not the final thing, but the last thing I'm going to talk about is how does linkage confound your ability to identify causal loci? As that original figure from Schlotterer showed, if indeed you have a rare variant in your population that's then the focus of selection, it's going to carry with it linked alleles also, and therefore you could be looking at a SNP that's really not at the causal locus, but just very tightly linked to it. So we wanted to know how far on the genome might linked variants be, but responding or being picked up by FLAM. So we did that since we don't have a specific genetic model we're using in our simulations. We did that by creating different degrees of correlations between the causative loci and the differentiated non-causative loci. And this was done, you can read the paper, how it was done by creating similar sets of subpopulations that showed close variation. So we had correlations that ranged, of course, from 0, which was all our previous simulations have been at, all the way up to, I think it was 92 or something. And what I'm showing here is for populations of size 20, how often we picked up the strong causal loci versus the non-causal differentiated loci. And you can see as the correlation, which of course linkage creates a genetic correlation between loci, as the correlation increases, there, in other words, as linkage gets tighter, the non-differentiated locus, it is more and more likely to be included in your sparse set as the causative loci. Now, when we had correlations below about 0.4, then we got fairly good separation, pretty close to what we get with 0 correlation. If you look in Drosophila, correlation of 0.4 corresponds to a level of linkage disequilibrium, which can be found in a region of roughly 10 to 100 kilobases in the Drosophila genome. So this method isn't going to do much better than getting you into maybe a plus or minus 50 kb region for where the actual causative locus is. Okay, so those are the basic properties of a FLAM, and just to summarize, again, the things that affect the ability of this technique to work are, one, the level of phenotypic variance on the phenotype you're measuring. Of course, the number of populations. So 10 populations, 5 controls, and 5 selected populations, which 30 years ago we thought was great, is really on the low side of what you'd want to use for these kinds of genomic studies. 60 to 100 would be much better, but as everyone who's done this work realizes, that's an enormous amount of work, at least for Drosophila, to have that many replicate populations. And lastly, that the causal locus might be within a region of plus or minus 50 kb of the SNP that you find. So there really are just regions that we're going to be able to identify, which can include a bunch of genes. Certainly in Drosophila, that's the case. So now I've already shown you some phenotypes that are differentiated. So what I want to do now is show some applications of the technique to some of our genetic data. So I didn't show this phenotypic data, but I mentioned it, that in the urea-selected populations, we have a phenotype called the larval feeding rate, which is slowed down in those populations. And so we had a set of 40 populations, 3 that were subject to urea selection, and let's see, what's the number, 2, 4, 5 that were not. And we we have genomic data for all of those 40 populations, and of course measured phenotypes, feeding rates for them. And this is what the output from PLAM looks like. And I'll just sort of summarize it here. So these figures over here have, for instance, the SNP location. And I should say, when we did this, we did some pre-filtering of our genetic data. That included, one, only using differentiated SNPs. We didn't look at everything because that just will slow things down. And then we looked at regions of plus or minus 50 kb to find a SNP that has the greatest level of differentiation. And these windows of plus or minus 50 kb were not overlapping because, as I said, we can't distinguish SNPs closer than that. Plus, if you add too many very close SNPs, it'll confuse PLAM also. So here are the genetic locations of the SNPs. Here, the big integer in each of these graphs is the number out of 100 permutations of the genetic matrix that locus or that SNP showed up in your sparse set. So this one that I'm looking at right now, 2L138, yada, 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 showed up 99 out of 100 times. So it's clearly an important player here. The x-axis is the frequency of the nucleotides from 0 to 1. And then the y-axis is a relative measure of the contribution to the phenotype. So you can see these are the step functions I mentioned that will be estimated by PLAM. For this locus, it shows a fairly flat contribution, but as soon as the SNP frequency gets pretty high, you get a big jump here. And that's similar data for, these are all the SNPs that satisfy that 50% criteria I mentioned earlier. Now, the other thing we can do with these data is, once we have identified this sparse set of loci that PLAM's picked out, we can take our data of 40 observations and we can divide it into a testing set of, let's say, 36 populations. I can re-estimate all the parameters of these step functions on that test set, and then I can predict the phenotype of the 4 populations that were left out. And then I can repeat that with a different collection of 36 and 4. So in that way, you can look at the relationship between the predicted feeding type, phenotype, which in this case is a feeding rate, and the observed feeding rate, and ask how closely do those match. And this figure on the right shows you that relationship between the observed feeding rate in these 40 populations and the predicted feeding rate. And the correlation between these observed and predicted feeding rates is 0.89, which is really good. And I should say, other large studies like this on human data, which have, 15,000 people that have different diseases, where they try to use really complicated genetic models to predict who will have disease and who won't, they might be doing pretty well if they get correlations like this of, 0.35 to 0.4. So So this is a pretty impressive correlation. Here we have similar results, but now we're looking at viability, survival in that urea food, again, for that same collection of populations. And we're not going to go through the individual loci, but you can see from this that correlation on those test sets, so we don't have a bias estimate of correlation, is 0.96. Again, pretty impressive. And of course, we've done lots of phenotypes this way. These last two slides are from Kasia Bittner's thesis work. These are unpublished, but soon will be published. The next thing we did, this is Thomas Barter's work, is we actually did RNA sequencing on these same populations and decided let's use the level of RNA at different loci as a phenotype itself. So in these 20 populations, because again, there's 5 replicates of each, ACO, CO, up to CO, we measured, we took samples at day 21 from egg to measure the relative abundance of the transcriptome. And you might remember from the phenotype data, at that age, there's a big difference in adult mortality. So now what we're doing is we're going to take genome sites, that is candidate genomic sites that we know are differentiated between these 20 populations. And again, the A's versus the C's show the largest level of differentiation. And I see that I mistyped this. This should be AO, not CO, because I have CO now twice. Anyway, it's ACO, AO, NCO, and CO. So we measured these at day 21. We got transcriptome samples. So what we're going to do, whoops, I skipped ahead. What we're going to do is take the genomic sites that are differentiated between the 2 A's and the 2 C selection regimes, and then we're going to use those as the input data to predict the transcriptome level. And here we're only going to look at genes in the transcriptome that are significantly different between the A's and the C's. And we're going to see which genomic sites can predict via FLAM the level of transcriptome in these A and C populations. So this is lots of applications of FLAM, and we get something like this. At the very top here are different genomic sites among those candidates that are differentiated in the A and C populations, and that the approximate locations are on the bottom of the x-axis here. And there's also color coding to each arm of the Drosophila chromosome. And then this collection of genomic sites were used to predict each one of the transcript levels that were differentiated. They're shown on the bottom here, at least their location is. And one of the things that we see is that there are typically many genomic regions that are relevant to the prediction of transcript levels. Now, that doesn't mean that these regions are specifically regulators of the genes, but somehow they're involved in that transcript abundance. At least that's what FLAM is telling us. We also see in this particular figure, it seems like more of the genomic sites from the X chromosome and 2L seem to be involved, but quite frankly, that's also almost certainly due to the fact that more of our candidate genomic sites are on the X and the 2L region of the second chromosome. So our overall take from this talk is that that we think this type of statistical learning or machine learning, whatever you want to call it, can be a really useful tool in the era of large genetic samples where, and especially experimental evolution, where we have a superabundance of genetic data on a fairly small sample of populations. And we think this technique is going to be helpful for trying to sort through that kind of data in the future. So I think I'm just about at 45 minutes, so I will stop here and entertain questions.
Thank you, Professor Mueller. I think I would like to start. There are a large number of questions, but I would like to start with a comment from Amitabh, because I also resonate with that comment. I like it. So Amitabh's comment is, it's so nice to see larval feeding rate as a test run of SLAM on real data. I can appreciate that. It's so nice to see that.
We've all had our issues with feeding rates, and it's a complicated but interesting story. Still a lot more work that can be done on it, but certainly with this kind of genetic data, we weren't going to skip that for sure.
So first question, why do the CO and MCO seem to be so much more tolerant to urea than TSO and AUC populations?
The most honest answer is I don't know, but let me just add the following. What we do know is populations that have been strongly or mildly selected for longevity, so the O populations are the ones whose age at reproduction has been pushed the most. The COs are pushed out, certainly not as far as the Os, but what we see in many of the populations that have been selected to reproduce later in age is that they tend to have more stress tolerance than the early reproducing populations. Now, 20 years ago, the kind of stress tolerance we were looking at is desiccation, starvation resistance, where the O's had increased in both of those quantities relative to the B's, the early reproducing population. It appears like they also seem to have a better ability to withstand urea, although not as well as the urea selected lines, but certainly better than the control line, AUC, which is on a 21-day life cycle. So that's the best I can do. We really haven't followed up on this. The data that I'm showing you on urea, larval urea tolerance for those NCO-COs, that's all recent and unpublished. So we certainly haven't followed up on that at this point in time.
Okay. The next question is about the FLAM method itself. Does how well the FLAM method work depend on the proportion of SNPs that are differentiated and have an effect on the phenotype versus the proportion that are differentiated and do not have an effect on the phenotype?
The answer is I don't know. So we used the set of fixed numbers that I showed, and that's a really good question. And I should say, the work that I showed there for the simulations, and I didn't show all of them from my paper, we're talking about a month or more of simulations straight. So it's a lot of computer work, but this is certainly something that could be addressed. I mean, it's just a technical problem, but it's not something that I've looked at. So that's an interesting question, and I don't have any natural intuitive feeling that I can just throw out there. And if I did, I probably shouldn't anyway, because I think most of our intuitive feelings, especially about statistics, are usually wrong. So yeah, I don't know the answer to that, but that's something we could address in the future.
Okay. The next set of questions are about the real data from the fly populations. Right? Are there among replicate population within selection regime differences in the loci that you pick up as causal for any phenotype?
So what happens when you use FLAM is you don't apply it to just a single population. You're applying FLAM to all of your populations. That is, for our urea work, we had 40 different populations. It would be like saying, well, I have one fly that weighs, a milligram. What can I say about that one observation? And the answer is nothing. You need to have a large sample. So, and in fact, having samples that show differentiation is better than not, because if they don't have differentiation, then that means the experimental error on your measurements are likely to obscure small differences between the populations. The best thing to use for FLAM to work would be a sequence like the development time data we have, where we have 10 populations that are very fast developing, 10 populations that are intermediate, and then 10 populations that are very slow. That's actually the best kind of data for FLAM to infer what's causal, assuming that the same set of genes are affecting development time in all those populations, which someone could argue may not be. But anyway, so you can't do FLAM on a single population because that's a single observation. So there's not enough information there to do anything.
Okay. Do you have some feel for how much error variance you have in the real fly populations and how it compares to the values you used in the simulated data set, which messed up your detection?
Right. So we certainly do for the range of variation in SNP frequencies. So we have a lot of information about how different those are. So the ranges that I use were ones that are not atypical of the genetic data we have. So on that case, or for that particular problem, we certainly have some relevant information about what are a range of SNP frequencies. And I should say most of the, just for people that are wondering, a lot of the differentiation we see genetically between selected lines aren't due to like 0-1, where one population is fixed for one variant and the selected lines are fixed for a completely different variant. Most of the changes we see are where we have a polymorphism, which is moved from one level to another. So from a low frequency to a higher frequency, but not fixation. And given some of these populations have been under selection for hundreds of generations, it's not a matter of time, right? Oh, well, you just waited 10,000 generations, they'd be fixed. I mean, they really seem to have gone to different equilibria. So we have a lot of these things called soft sweeps, which molecular genomic people talk about, as opposed to what you typically see, of course, in bacteria, where you get a mutant and it sweeps through the population to fixation. That is not a characteristic of the adaptation or the genetic changes seen in these populations. So for genetic data, we use variation in SNP frequencies that we find in our own data. For phenotypes, this is a made-up phenotype, but you can see from the phenotypes that we have incredible differentiation, like many standard deviations differences between populations, and that was roughly, we use those as guides also for generating the phenotypic differences in our simulated populations. So there were rough boundaries that were set by real data, but in the end, the data was cooked up by doing sampling under the conditions and the limitations that I just described.
Okay. Next is a set of two questions, one more question, one a follow-up. On the predicted observed urea viability plot, why does the relationship level off at the extreme values? Is it because the test data are less likely to include extreme points and the middle points?
Yeah, so if let's see, happening here is we obviously, you've got a series of populations that had this difference in ability, and I would have to actually look at the data to figure out how that happened, because what we're actually plotting in this figure is, because we had so many populations, we couldn't run this viability study at the same exact time. So we had one population or one selection regime that was used in each one of our temporal samples. And so these viabilities that I'm showing in the FLAM curve were basically the viability relative to that standard. Did they survive better or worse? So I have to look back at the individual populations to see how that happened. But yeah, you're going to get a straight line, as we did at the extreme, where you have a bunch of populations that had the same value, and then populations that survive at all in the urea. That's only one endpoint there, where we could have had a couple that did really terrible.
Yeah. Professor Mueller, I have one basic doubt. In all your selection experiments, body size and body weight are not considered. Why?
Well, we actually do have, I didn't show you here, one of the experiments we did with the urea lines and the control types of populations is we did a large experiment measuring the growth of larvae through all the larval stages up to adults. So I have growth curves when we measured the wet dry weight of larvae basically every 6 hours, from about 24 hours to about 110 hours. And we actually estimated growth for that data. And we had the adult dry weights also. So we do have information on that. But turns out that the growth trajectories of the larvae are different, but aren't as different as feeding rates or survival. So when we did this type of FLAM analysis on them, the correlation was much lower, which is still not bad, but it's not nearly as dramatic as at some side. Just didn't get the show. Yeah.
Thank you, sir. Professor Mueller, I have nostalgic memory to share with you. In the early 70s and late 70s, I started my research career. Then our model was Professor Ayala. We did a lot of competition experiments, and such a prolific writer and experimental biologist. He was our model.
Yeah.
Ayala, who was your doctoral father.
Yeah. Okay. Very good.
So, and believe me, whatever reprint requests of those days, we used to send it by air mail without delay.
Oh yeah.
Those days, no internet, no email, nothing, everything by mail, by post. We used to respond. So, and when after doing the experiment, we were supposed to calculate relative fitness, adaptedness. I used to send the data to him. He used to do all statistics and send it to us.
Yeah.
He was a great teacher.
Yeah. No, Francisco was great. So it turns out that, Amitabh Joshi worked with me, actually both as a graduate student and as postdoc. And I've had to mail things to India, and see the terrible things that can happen in the mail. But at the first book, we wrote a book together in the late 1990s. It became easier to send book chapters via email, and we wrote the whole book. He was in India, I was in the U.S., exchanging book chapters without having to mail anything, and it was fantastic, because I think if we had to mail each other things, it would have taken twice as long, if not longer. Yeah.
Thank you, sir. Thank you. Thanks for
Thank you, Professor Ramanathan. Thank you.
Finally, Prasad, we are here. Prasad, Vidya, myself are all, we are in the school of our Joshi.
Yes. Right.
He's our philosopher and guide for all of us.
Yeah. Well, Amitabh was my first and still best student. So, yeah.
Thank you.
Thank you.
Professor Mueller, I think we have addressed all the questions. I would like
to
thank you so much for a fantastic talk. This part of your research is something that I had not read about. So this is really, really nice to learn about it. Thank you so much.
My pleasure.